Thank you very, very much for your kind invitation. It's really a great pleasure. I've been saying to everyone how much I learned personally because, you know, we physicochemists are a bit dumb, you know, so we play with proteins, but, you know, we lack the context, you know, inflammation, all kinds of things. So thank you all for, I have been really learning more than I can, so, and, you know, thanks a lot. So, yeah, I'm going to talk about the infectious properties of protein assemblies involved in neurodegenerative diseases. So this is, so I don't know, it's not showing. Interesting. No, it's not the embargo. Okay. See, it wasn't embargoed. So I will start by introducing protein folding and misfolding, so just in 1 minute. Then talk about prion disease in mammals because, you know, this defines how we see the aggregates that we are forming in test tubes. Talk about the molecular basis of prion propagation and amplification. Then I will get into what we are mostly interested in other words, the propagation of these protein assemblies that we have, that are involved in neurodegenerative diseases. Talk about protein toxicities and talk about the strains. And in the second part, a sort of discussion part, I will tell you how I think we should interfere with the propagation of these fibrils from one cell to another. And I will sort of list what I think is important to document because I see things here as a way to interact with you guys, and you will see how much we interact with partners. And so I think we can bring in some knowledge, and you guys can bring in also some knowledge, and we can move ahead. So this is really a basic scheme. I'm pretty sure that you all are familiar with. So the proteins are newly translated. They populate folding intermediates. Some of these folding intermediates will fold into a native protein, which will, you know, oligomerize to form like quaternary structure, like hemoglobin, tetramers. Fibrils, like actin filaments. Crystal, like in the lens. You are watching me through your lenses. And in some cases, you do need a class of molecules, they are called molecular chaperone, to assist the protein to reach the native state. So let's forget now this. This is what is represented here. In some cases, the folding intermediates have the propensity to interact with like proteins. They then form fibrils. These fibrils are the signature of all the disease that we have been talking about during the last 2 days. From Alzheimer to Prion disease to Parkinson, etc. In some cases, it's degradation products that assemble into fibrils. So this doesn't happen only in the brain. It happens also in other tissues, but we will be focusing here on the brain with tau, A-beta, alpha-synuclein, Huntington, and other proteins that are involved in dementia. So as the misfolding and aggregation of newly synthesized protein, the unfolding of native polypeptide following a stress, for example, and also the incorrect elimination of protein, as the probability of having these processes not going right increases with time, these diseases, which some of us call conformational diseases, are associated to time, in other words, aging. And while this colleague is saying something right, in other words, birthdays are good for you because statistics show that people who have the most live longest. Birthdays are bad for you because the more you have, the more you are at risk of developing these diseases. So just an introduction to prion diseases. So prion diseases are naturally occurring diseases. There are a number of these diseases that affect human, cattle, sheep, etc. They occur naturally, but you can also induce the disease by contaminating mice, for example, here with BSE, and you see that the mouse is developing ataxia. And you are more familiar than I with the mice. This is the normal behavior of a mouse. The disease only occurs in animals expressing this protein, the prion protein, and knockout animals are sort of resistant to the disease. So here are some characteristics of these diseases which are sort of common, I mean, you are familiar with. The incubation time is very long. It's the case for the disease we are interested in. They are transmissible. The lesions are in the central nervous system. There is no major inflammation, no microorganism proliferation. You have the accumulation of this protein, PRP, in the central nervous system, and the central nervous system looks like spongiform. That's why they are called spongiform encephalopathies. And usually the amount of accumulated protein correlate with the infectious titer. What we know about the PRP is that it binds to cells, it gets internalized, it transforms the constitutive protein into an aggregated form. It propagates in the neuron, it moves in the axon, for example. It propagates between neurons through probably the synapses, but also through tunneling nanotubes. And there is a process of amplification, breakage of the fibril, etc. So how do we see, from my point of view, what is happening? So we don't know exactly because we don't have a structure for this protein. But what we think is that we are generating these folding intermediate that can interact either longitudinally or laterally with the same molecule, the same folding intermediates. As long as you have this oligomer, the oligomer is unstable because there are either longitudinal or lateral interactions. It's when you have longitudinal and lateral interaction that you stabilize dynamically, thermodynamically the assembly. And I represented things in a way so that you see that if you add a brick here, you create another site for another brick, and so you can grow indefinitely, basically. And that's what is represented here. You can break these fibrils by Brownian movement, and the more you break them, the more you generate ends, and they are growing from their ends. Please stop me if something is unclear. I mean, it's way more interesting to discuss than to just tell you what I think science is. In a test tube, you can purify the protein, incubate it. Not much happen at the beginning, so that's what we call the nucleation phase. During the nucleation, it's these oligomers and the first fibrils that are forming. We cannot detect them. As soon as the fibrils form, they will elongate by recruiting the protein and displacing the equilibrium toward the formation of this folding intermediate. And because we are working in a test tube, at one point, everything is assembled. So we reach a steady state. And you can look at what you have in your test tube using electron microscopy. So at the beginning, you have these small oligomers. Later on, here in the elongation phase, or at the end of nucleation, you see larger oligomers, some fibrils, and later on, beautiful fibrils. And we have been doing this with yeast prions forever. Now, if you take these fibrils here and dilute them in a fresh solution, what you abolish is nucleation, meaning that nucleation is the critical step, is the limiting step. And then you have an explosive assembly, elongation, and steady state, again, because you have a given concentration in your test tube. So it's a seeded process. So I was recruited like this misfolded protein to the field. After I read a number of papers, one of the things that struck me most is the work of Heiko Brack, who described, pattern of deposit during time. This is very important to keep the time axis in the brain of people suffering from Alzheimer's disease, from Parkinson's disease, and later on, people extrapolated to Huntington's disease. And we'll talk slightly about Huntington's disease. The other papers that really struck me and got me really recruited this time, so I can't thermodynamically even anymore leave the field, are these two papers from Patrick Brundin and Jeff Cordover. So we heard about these papers from the first day. So you remember that some patients were offered to have fetal tissue grafts in their brain, and these group were looked at the brain after the patient died, like 15 to 20, 15 years on average after the patient passed away. And what they found is that Levy bodies invaded the graft. So basically, what was in the old brain was invading the piece of the brain that was only 15 years old. So this sort of really struck me. And I used to meet Ron Copito and Patrick Brundin at meetings dealing with amyloids, yeast amyloids. So this is where we sort of discussed, and I decided to move to Ron Copito's lab for 6 months or so for a sort of visit where I could answer simple questions I raised with him. And also I interacted also with Patrick at the same time, but without visiting his lab. At that point, he was in Lund. So the questions are very simple, I'm a physical chemist. So the question is, are these assemblies Huntington, because Ron Copito lab has been active in the Huntington field, and alpha-synuclein, because Patrick is active in the alpha-synuclein field, are these assemblies infectious? So basically, the questions are really listed here. They are basic, So do these assemblies bind to the cells, which are represented here? After they bind, do they penetrate the cell? If they penetrate, do they induce the aggregation of the endogenous protein in such a way that they sort of amplify? And if they do so, can they propagate? What is their fate? So just a brief introduction. Huntington's disease is a genetic disease. So you have a person here suffering from disease. It was described by this colleague. It's an MD. Basically, you will not develop the disease if you have a limited amount of glutamine in this protein, Huntington exon 1. The patients will develop the disease when they have more than 37 glutamine within the Huntington gene, the Huntington protein, or a fragment of this protein which exists in real life. So there is this threshold here between 35 and 40, and what is happening is what is shown here. With 40 glutamine, the protein start aggregating in neurons while it's soluble before, and the more you have glutamine, the more you have aggregates. You have neurodegeneration, and there is a relationship between the age of onset and the number of glutamines. The more you have glutamine, it's a simplified way of presenting things, but the more you have glutamine, the earlier you will develop the disease. So Huntington has many functions, but I'm not interested that much into the function, although they are very, very important, but I'm interested essentially by its aggregation propensity. So in this fragment, it's a gigantic protein, and in the fragment that is detected in life, in vivo, it's a degradation product. You have a very abnormal amino acid composition, like these polyQs, they're followed by polyprolines, and then with proline-rich region, and so on. So you can express this protein. It's not as gigantic as this one. And when you express it, and you have different version with increasing number of glutamines, and you put this in a test tube, what you see is within this time frame, with 15 glutamine, the protein does not assemble. With 25 glutamines, the same. With 35, you have a lag phase of about 500 minutes. And with 45 glutamines, you have, at exactly the same concentration, nearly no lag phase. And it's concentration dependent. The more the protein is, the higher the concentration is, the shorter is the lag phase. And you can look at these objects at the early stages of assembly. So you see basically mostly oligomers. Some are sort of flat, like bidimensional crystals. And later on, you have these bundles of fibrils. So first question, you remember a sort of basic one. Are these aggregates capable of binding to cells? I'm certainly not the first one who showed this. So to deal with this anyway, I grew cells, I suspended them, distributed them in test tubes, and I labeled the fibrils with a fluorophore, extrinsic fluorophore, added them to the cells, and went to a cell sorter, a FACS, and looked at the fluorescence of the cells before and after adding the aggregates. And you can see that all kinds of cells grown in many labs bind these aggregates. If you are quick enough, you can do even kinetics of binding. So you add the aggregates and immediately put the solution in a fax machine. You can see how fast I am with the first time point. Still though, in less than 2 minutes, everything is over. 100% of the cells had the aggregates. So what happens after? Yeah, I don't look like that, but I'm fast. So what happens after binding?
With this experiment, you don't know whether it's just bind to the membrane, to the external membrane, or whether it is.
Exactly, yeah. So what happens after binding? So to answer what happens after binding, we generated this cell line, which is expressing a subpathologic form of Huntington fused to a fluorescent protein, cherry FP. And we exposed the cell line to either assemblies or a solution of Huntington with 18-glutamine, so it's subpathologic, or preformed fibrils in test tubes made of Huntington, a version of Huntington with 51-glutamine. It's not poly-Q, what I'm showing you. It's really Huntington exon 1. And you can immediately see what's happening. The protein is expressed in the cytosol. In this case, the fluorescence remains even. In this case, you have a reorganization of the fluorescence with foci. And you can quantify all this. You can count the cell and the number of cells with foci. And you can see that we can get up to nearly 60% of cells which have foci, meaning that the aggregates bound and got internalized. They reach the cytosol in a way or another, and they are recruiting the endogenous protein. So all this is sort of artificial. So I thought that I would do something in vivo. And by that time, I thought that cell cultures were in vivo. I learned 2 years ago that it was not. So I was depressed at one point. You should remove this from the Anyway, so I thought I was doing some in vivo stuff. It's in vitro. So this experiment I did with my own hands, so I'm really proud of this experiment. So what I did here is I generated 2 cell lines. This one you are familiar with. It's the Huntington Q25, subpathologic Huntington exon 1. It's soluble. I showed you it's expressing the protein that is labeled with a cherry FP. And I made another cell line which expresses a Huntington that is aggregation prone, that is fused to a GFP. So when you look at them in the microscope, you see aggregates in these cells, nearly 100% of the cells, and no aggregates in these cells. So the idea was to grow these 2 cells and determine whether these aggregates were moving, the green aggregates were moving from the green cells to the red cells, in which case the green aggregate would recruit the endogenous soluble protein in the red cells, and the aggregate would appear orange. And because I'm impatient, I organized to have these cells puromycin resistant so that I can kill these cells and sort of activate things if things don't go fast enough. So here is the result. So here we are counting the cells with expressing the cherry fluorescent protein with foci. Even when I don't add puromycin, I reach like 12 to 15% of cells which have green aggregates, red cells that have green aggregates, and the green aggregates have seeded the endogenous protein, so they look orange like this. And you can see at the beginning, you know, the cells expressing the aggregated form of Huntington and the soluble form of Huntington grown together. Now, if I activate things, in other words, kill the green cells, you can see that we can get up to 55% of cells that are expressing the soluble form that recruited the aggregates that are green coming from other cells that we killed and that are being used to seed the aggregation of the endogenous protein. So we'll move now to Parkinson's disease. So Parkinson's disease, you are, again, way more familiar than I with Parkinson's disease. All I wanted to say that it's the characteristic of this disease are these Levy bodies and the Levy neurites. And Maria Grazia showed that, in fact, these aggregates were made of this protein alpha-synuclein that we have been talking about for a while. Was this the picture that was taken for your presidential campaign? Okay. So alpha-synuclein. So alpha-synuclein is a sort of, we call it natively unfolded protein. In fact, it folds when it binds to the membrane, and it forms helices. There are a number of familial mutations that are associated with early onset Parkinson's disease. People like to sort of make like bits and pieces and domain within this protein. The protein assembles into fibril, and one can follow the kinetic of assembly, and you can change the concentration, check what you have in solution. And so far, we have these 5 mutations. Plus, if you overexpress alpha-synuclein in some familial form, the alpha-synuclein is overexpressed by factor 2, 3, etc., because of gene duplication, and these people develop the disease. So again, the same question that I asked earlier with Ron Copito that were asked again with Patrick Brundin. So the question is, do these assemblies bind to cell? The answer is yes. Whether the alpha-synuclein is monomeric, oligomeric, or fibrillar, the protein is labeled with an extrinsic fluorophore. It's green, and the cells you see. And once they bind to the cell, they are internalized, and they can recruit the endogenous protein and seed the aggregation of the endogenous protein. All this is done in vitro, in cell cultures. So I thought, yeah, because I thought that at that point I was doing in vivo. I thought, yeah, let's move to a more relevant cell that is in vivo, you know? So it's neuron, primary neuron. So what we did with colleagues in Stanford is use these microfluidic devices where you can grow a neuron in such a, primary neuron, and you can use these gradient pressure to get the cells to expand their axon through microgrooves. Each microgroove accommodate like anything between 1 and 10 axons. So you have the soma, the cell bodies on one side, the axons on the other side, and you can label these with either choleratoxin or calcine or whatever. And the idea here is to look at the movement of these particles after they are taken up. So what we did is we added preformed fibrils on one side, and we looked within these microgroove whether we could detect aggregates. So the aggregate cannot be in the microgroove unless it is in an axon, because at the same time that we add the aggregates, we reverse the gradient pressure in such a way that nothing can go from here to here. So you can see here an aggregate, and there are a number in the axons. When you do this, you can also track these aggregates and tape them, you know, and do chymographs. So here you have an aggregate that is tracked with a chymograph, and you can see how many are moving, you know, so it's not like a rare event. From these movies, you can derive these chymographs, and you can see that the aggregates are moving retrogradely and anterogradely, and in some cases, they move, stop, move, and stop, etc. With different speeds. But still, overall, the speed at which they move is compatible with fast axonal transport. Then we wondered whether after moving, these aggregates can be transmitted to other cells. So what we did is exactly the same experiment, add the aggregates here, but in the axonal compartment, we had other primary neurons sitting in the axonal compartment. And what we saw is that if you add the aggregates here, wait 24 hours, you will get the aggregates to the secondary neuron, if I may call them this way. So we are constituting like a sort of minimal network where you have one neuron giving to a second neuron. And some people now are building a more complex system where you have 3-chamber and also systems where you can stop propagation between chamber 1 or 2 to 2 or chamber 2 to 3 and injure the neuron so that you can answer to questions such as, do you need to transfer everything through the axon, or you can just injure the cell and release the aggregates in the medium, and would they propagate, and so on. Now, so to summarize this part, alpha-synuclein assemblies, but also Huntington, are binding to primary neurons. They are transported through the axon. They are internalized in second-order neurons. This is essentially what is schematized here. And if you don't use these microfluidic, but just grow these neurons on a plate and look at them, it's fascinating. I mean, look how much they are trafficking between cells. You only see here the fluorescent exogenous aggregates. So this, to me, again, because I have a sort of simplistic way of thinking, sort of suggests that these aggregates are propagating as prion do. Yes. So we did also the experiment the other way. So we added the aggregates to in the axonal compartment, and we saw that it goes up to the dendrites, and they are propagating to second cells. And we did it because, again, we are physicochemists. So this is work that will be, I hope, published soon. We quantified all these transports for different aggregates. We'll discuss this if you wish. So let's get back to the schemes and the patterns described by Braque. Again, it's impressive and it says something. It says that the aggregates are first appearing in the olfactory bulb and in the brainstem, basically. I'm simplifying things again. So with Patrick, we decided to see whether our exogenous aggregates can propagate. So what we did first is inject the exogenous aggregates in the olfactory bulb of mice and rats. And the question is, are these aggregates or different form of the protein moving to connected regions? And as you can see from this scheme here, the monomeric, oligomeric forms do move within a relatively short time. You know, it's like a maximum 3 days here. The fibrils do not. But there is a follow-up on this study, and Patrick showed that if you wait sufficiently, then you have the fibrils propagating, and he talked about this at the ADPD meeting, if you remember. And this is just to show you some histology. And exactly. And this is where he compared our fibrils to Virginia Lee's fibrils. And when he said that, in fact, in Virginia Lee, he has like mixtures of things, and he cannot really relate things, while in our case, our fibrils, we reproduce really disease. Yeah. So with Jiayi Li, who was a collaborator of Patrick Brundin, we then injected the aggregates in the muscle wall of the intestine, and we were counting on the vagal nervous system to sort of propagate, to take up these aggregates and move them up, and that's what happened. So we injected fibrils, oligomers, monomers, etc., but also PD brain, aggregated alpha-synuclein from PD brain. They are all labeled with extrinsic fluorophore. They were injected in the intestine, and we could follow over time their movement, you know, upward to the brainstem. And you can see here that within 6 days, our fibrils and the PD lysate reached the brainstem, meaning that what we observed in these microfluidic device is happening also in living animals. So this work is summarized here. So these species are propagating from the olfactory bulb to interconnected brain regions, from the gastrointestinal tract to the brainstem. Others injected the aggregates into the muscle and saw that they find them in the central nervous system. So all this supports Braque's hypothesis. In other words, we have a factor that is propagating into the brain following defined networks. So when you inject these aggregates to recipient animal, model animal, they are toxic. The mice will die, and they are in toxicity, and they are sort of comparable to when you inject aged brain to these mice. So the question is then, why are they toxic? Remember, I'm a physical chemist, so you get back always to your bad habits. So why are they toxic? When you ask this question, you get into this field where people oppose. Some say it's the oligomers that are toxic, and some say that it's the fibrils that are toxic. And I have to say that the physicochemical data for oligomer being toxic is really weak. So, nonetheless, in my opinion, it's totally irrelevant to compare the toxicity of oligomers and fibrils as a function of the initial alpha-synuclein or Huntington or whatever concentration. Why is that? Because oligomers are made by very few molecules. It depends on the molecule, on the protein, but they are made on average between 4 and 8 molecules, while the fibrils are made of thousands of molecules. Plus, the fibrils are very heterogeneous in length. So again, it's totally meaningless to say oligomers are more toxic if you compare the toxicity of fibrils and oligomers at one given concentration of alpha-synuclein that is monomeric. So to compare toxicities, because the cell is seeing particles, you need to define at any given concentration of protein what is the actual concentration of oligomer in micromolar and what is actual concentration of fibrils in micromolar. And as I said, the fibrils are heterogeneous in length. So to sort of homogenize them, we started sort of sonicating them, not very actively, but sufficiently to make them relatively homogeneous in length. So then you can use machines that exist since the 50s and even before, which are called analytical ultracentrifuges, which allow you to estimate the molecular weight of these particles. When you do that, you determine that, in fact, oligomers of alpha-synuclein are made of, on average, 8 molecules, while the fibrils we are using in this study are made of thousands of molecules, based on their molecular weight.
That's presupposing that you have, say, a monomer, like a single, you have fibers, you have many interaction sites, so it would be highly cooperative, even though, from the point of view of the monomer, you have a different concentration in the fiber. But you're assuming that the interaction is like a single molecule, so a fiber is a single molecule, and an oligomer is a single molecule.
Mm-hmm. It could be, yeah, you are right. It could be cooperative, but I doubt it is because, in fact, alpha-synuclein in an oligomer and in a fibril is not exposing the same surfaces to the environment. Yeah. Yeah. So, yeah, you will see how I'm presenting things. So I'm relatively cautious. But what we can conclude from these experiments where we exposed cells and counted them after 24 hours to increasing concentration of fibrils or oligomers is that with 4 nanomolar fibrils, you have like 50% of cells dying. It's not an MTT assay. We are just counting the cells after 24 hours. And with 1 micromolar of oligomers, you have nearly 100% of the cells surviving. So I think this is sufficiently strong to say that fibrils are more toxic than oligomers. Could be. But if you get into this longer, how can you guarantee that the oligomers are not reorganizing on this mosaic, this fluid mosaic that is the membrane? So, I mean, nothing is perfect, but again, comparing the toxicities of oligomers and fibrils at concentration which are totally irrelevant, soluble protein is meaningless completely. Because again, the cell, I consider that the cell sees a particle. And by the way, the people who present oligomers are as sort of ionophores also reason in terms of particles, by the way. So why are they toxic again? So to answer this question, we moved again to really physicochemistry. Oh, sorry. We produce lipid vesicles of known composition. We exposed them to these fibrils, and the first thing we did is to layer them into density gradient and spin. And when there is an interaction, you expect the fibrils to go up like this guy, because their density goes down. And when there is no interaction, the fibrils remain where they are. So what we saw is that, for example, with alpha-synuclein, this is the control. There is nothing in the top fraction. With PG vesicles, you see that the alpha-synuclein is redistributing. It's fibrillar alpha-synuclein. So there is an interaction. We then use this assay that many of you know, where you encapsulate calcine, a fluorophore, in lipid vesicles. So this fluorophore auto-quenches. And when it's released after, for example, membrane permeabilization, the fluorescence goes up. And sure enough, you can see here that when the fibrils are interacting with PG vesicles, you have permeabilization, while with PC vesicles, where there is no interaction, there is no permeabilization. So let's now move to cells, other vesicles in my eyes. We filled the cells with a fluorophore that is sensitive to calcium concentrations, and we exposed these cells to fluorescently labeled alpha-synuclein. I don't know if you see, but it's red, and you can see it here on the cells. And we recorded over time what happens, and you can see that the fluorescence is increasing. You can measure individual cell or many cells. And what you see is that there is an increase in fluorescence, meaning that the membrane are being permeabilized by the fibrils. If you add EDTA in the medium, you will not have this increase, meaning that the calcium is coming from outside. And this flux of calcium activates caspase 3, and you have this programmed death of the cell. So is this specific to alpha-synuclein? The answer is no. You can make Huntington exon 1 fibrils, make them homogeneous in length, expose the cells. As you can see, there is toxicity associated to fibril, not to the oligomeric form. But there are differences. For example, Huntington exon 1 fibrils will interact with PC, PS, PG, whatever vesicle. They permeabilize these vesicles, they permeabilize the cells, and you have calcium waves. So as to summarize this part, we think that it's the fibrillar form of these assemblies that are most toxic. This doesn't mean that oligomers are not toxic. We think that they are toxic because they permeabilize the membrane, they have some specificity, they alter the calcium homeostasis, and this leads to cell death. So, one other thing that struck me is that, and again, you are more expert than I, the aggregation of alpha-synuclein leads to distinct diseases. And when you say this to a sort of protein chemist, a crystallographer, or whatever, he will say, interesting, because it sort of remind me that you can take the same protein and crystallize it in different forms. And this is exactly what happens when you assemble alpha-synuclein. This is not our work, but this has been published many times in journals. So I picked up a couple of examples. When you assemble alpha-synuclein in a test tube, you don't have one single form in the electron microscope. For example, in this study, you can see 3 forms. Some are soft, some are twisted, some are right-handed, some are left-handed. and the pitch is not the same, meaning that this is heterogeneous. And for me, as long as it's heterogeneous, you can conclude nothing about these properties. Because it could be that it's a minor species that is creating the largest effect. From this study here, where they use cryo-electron microscopy, so it's not negative staining and some kind of artifacts that can be due to negative staining, you can also measure the width of the fibrils, and you can see that the fibrils have different widths, meaning, again, that they are different. And I have to say that in my lab, when we started working with alpha-synuclein, we had exactly the same results, so we are not better than these colleagues. Who have been, again, in the field for over 20 years before us. So at one point, I got really mad because the results were like sort of with large error bars. And I said to one of my previous students, he did his PhD with me, and he got a position, so he's an associate, he's my principal associate. I told him, look, we are doing biochemistry, you just go away, revisit the whole system. I don't want to see you before you get back to me with homogeneous preparations. Yeah, see, I can be very harsh. So Luc Bousset worked for a couple of months, and came back to me with conditions where you have still mixtures and conditions where you have 100% homogeneous particles. The interpretation of this is that I cheated on you. You have this folding intermediate here that is populated from this whatever protein that assemble into these fibrils that I showed you earlier. But you have other folding intermediates that can be populated and assemble into different fibrils. Okay? The secret is to have 100% homogeneous fibrils. If you have a mixture, you can conclude nothing. If you have 1% of this, and this is toxic, and this is not, then you will conclude it's toxic and it's not. So here are the evidence that these fibrils are different. So Luc generated several strains. We call them this way, and you will see why we call them this way. I'm showing you here two forms. Already in the microscope, and again, it's not one fibril that I'm showing you several fibrils. You can see that these fibrils have nothing to do with these. And here we are observing the fibrils in the electron microscope. These are flat, these are like thin. Now we subject, so all the techniques that we implemented come from the prion field. We learned a lot from the prion field and in the yeast prion field. So we subjected these aggregates to limited proteolysis with proteinase K. You can see that the degradation pattern here has nothing to do with the degradation pattern here. Meaning that the cleavage site for the protease that are present here are not present here, or vice versa, which gets me to the surface properties of the fibrils. Then we asked Paul Henning-Jensen for his conformational antibody, the FILA antibody, and as you can see, a pan-alpha-synuclein antibody recognizes both forms on this slot plot, while the FILA antibody recognizes this form, but not this one. Again, the surfaces are different. The epitopes are not exposed the same way. And it's all or nothing here. I mean, you know, it's pretty straightforward. It's exactly what we find. Let me show you. So then you do, you know, all the techniques that we are used to do. For example, X-ray fiber diffraction. So you align the fibrils in a capillary, you expose them to X-rays, and what you see is that the images of this form and this form are different. In both cases, you have this 4.7 angstrom reflection, which is the distance between two strands. It's characteristic of amyloids, but they are not the same. Then you do solid-state NMR. So this is not a psychological test, you know, here. Don't try to see anything in there. It's, or if you see something, you have to tell me. This is solid-state NMR. This to get this, we had to produce grams of alpha-synuclein in a homogeneous state. And this panel here, I refer to, I think, Roger's presentation, took us 2 years, okay? So it's not like just a small thing. So from this, you can determine the secondary structure content of your fibrils. And this is what is represented here in the bottom. The form that we coded blue has nothing to do with the form we coded red. So both have beta strands. This is what these arrows mean, but they are distributed differently, okay? And to get back to your question, notice that after amino acid 100, we have no structure, meaning that it's flexible in both cases. But what's also interesting here is that in one form, the structures start at amino acid 1. In the second form, it starts at amino acid 15. So there is also flexibility to some extent between different forms with the N-terminus, meaning that we can design tools to detect, one form versus another. Also, what is really funny is that then you can go back. I mean, if we express grams of wild-type alpha-synuclein, we can probably expose grams, if not more, of the mutants, one of which we characterized with colleagues at ACM in Paris. If you have the G50, all the mutants can assemble into fibrils, and I will call this from now on ribbons, because they are flat. A part 1, and this is sort of the demonstration that our structure is right, it's the G51D. No, sorry, it's not the A30P, sorry. Why? Because if you change an A to a P, you create a 90-degree bend, and it's just located right here. Okay, so you break this strand, and you cannot form any more ribbons. But you can still form fibrils. And why is that? Because we are in a flexible region where you can accommodate a bend of 90 degrees. Yeah, so this we don't know yet, but this needs to be documented, and this is what we will be discussing in the second part of my talk. So basically, we can imagine and extrapolate what I'm showing you here to what might happen in different conditions in different cells in our bodies. So depending on the conditions, we could form fibrils and ribbons, because here the conditions are neutral conditions, physiologically relevant conditions. But we don't have yet this demonstration that if we get aggregates from MSA, PD, etc. patients, we can amplify different structures, and we are working on this. But we sort of finished the demonstration the other way around, and I will be showing you this. In our hands, it's no more toxic than the wild-type protein.
But it's because the structure is different.
Mm-hmm. No, but I'm.
This is physiological change. The other are structural change.
So what I mean is that if you assemble A30P or wild-type into fibrils, because you cannot form ribbons anyway with the A30P, they have overall the same toxicity in our hands. Let me show you. In what I thought was in vivo. Okay, so to answer this question, what we did is some functional characterization. So here again, you have cell, the same type of cell is neuroblastoma cells that are exposed to fibrils or to ribbons. And you can see in the facts that the binding is sort of similar, the kinetics are similar, but fibrils bind to a higher extent than ribbons to the cell. And when you image this by calcium, with a sensor of calcium, you see that the fibrils permeabilize to a much higher extent the cells than the ribbons. So that's one explanation for a higher toxicity. And if you now look at cell survival or MTT, by MTT assay or counting the cells, I mean, you can look at the paper. There are many assays that we did. You can see that the fibrils in red are way more toxic than the ribbons in blue. But what's really interesting is their seeding propensities. So if you expose these cells that we generated that express an alpha-synuclein fused to a cherry FP, the protein is soluble, as you can see here in the control. If you expose them to very limited amount of fibrils, you can see that after 18 hours, most cells have these puncta, and they are orange because they recruited the endogenous protein. Now, if you expose the same cells to 10 times higher concentration of ribbons, you barely see that many aggregates, meaning that the ribbons recruit the endogenous protein with less efficiency. They have a lower seeding propensity. You can look then to their persistence, which is another way to look at their efficiency of recruiting and resisting the clearance machinery in the cell. And you can see that the fibrils persist up to 60 days, after diluting, the cells and growing them, while the ribbons end up being sort of cleared after 30 days. But the most important thing is that if you now look at the resistance to proteolysis of the endogenous protein, so it doesn't have a molecular weight of 15 because it's fused to cherry FP, you can see that the fibrils are generating aggregates within the cells that resist proteinase K, while ribbons are not. Meaning that it's sort of imprinting, the exogenous fibrils are imprinting their own structure to the endogenous protein. So we thought that since this is not in vivo, we would move to animals. That's in vivo for sure. Maybe it's not relevant animals, but it is for sure in vivo. So what we did with vialebekelant is injecting these different fibrils, the fibrils and the ribbons, into the brain of animals. The fibrils are labeled, the exogenous aggregates are labeled with a fluorophore. And what we are doing here is ex vivo fluorescence imaging. So we are, looking at the fluorescence of the object after 20 minutes or after 7 days. And you can see that the aggregates sort of propagate within the brain within 7 days. We looked at the behavior of these animals which were injected, these aggregates. So we are injecting in one lobe and looking at the consequences in the other, in the other side of the body. And what you see is that when you inject these aggregates, 90 days after injecting these aggregates, but not the oligomers, you see a sort of physiological consequence. The animal is not using, so for example, here the animal was injected in the right hemisphere, and you can see that he's not using in the rotarod as much as normal the left but I don't know how to say feet. Okay, and the control is really inside the animal, so it should use 50-50. Then we looked, Viale and her colleague looked at the fate of these aggregates. So we are generating with the fibrils and the ribbons Levy body-like aggregates. But with the ribbons, not with the fibrils, we are generating fibrillar assemblies which sort of mimic Levy neurites. And this, I understood from the specialists, is sort of a typical, this sort of rod-shaped thing is something that sort of is typical of not Parkinson's disease anyway. Well, that's what I understood. And again, you can quantify all this. Then we looked at whether these aggregates go to the dopaminergic neuron, and whether they have the staining, the typical staining of Levy bodies. So we used this protein, P62, which is a ubiquitin binding protein. And you can see here that not only alpha-synuclein, the exogenous alpha-synuclein is phosphorylated, but it also is ubiquitinated. And when we looked at the oligodendrocyte, we had, GCIs. with ribbons, not with fibrils. So there are differences between the two forms in vivo. So then we did some electrophysiological recordings. So we saw some of these during the meeting. So we did patch clumping on single cells or on networks of cells using these MEA culture chambers. And you can see that that's why I keep saying that I never said that oligomers are not toxic. I say they are less toxic than fibril. But you can see changes as soon as you add oligomers, ribbons, or fibrils, not in the control. And the same is true for the recordings we did with the MEA, keeping in mind that we are adding at most 1 micromolar of alpha-synuclein expressed as a function of the soluble protein concentration, meaning that in the case of fibrils, it's like less than nanomolar concentration. While the cells, we know, other people measured the amount of alpha-synuclein within these cells, the concentration is 40 micromolar.
Because you have, I mean, in some cases you have your fluorophore, and in some cases you don't. I mean, you're using monomeric and cherry, so I mean, you don't have a intensity. It's not like where GFP maybe or the old.
Sorry, yeah, I wasn't clear enough. All these experiments are done with animals and with cells which are not transfected, so it's not expressing anything. We are adding the fibrils, but they are all fluorescently labeled, and they are fluorescently labeled exactly to the same extent. So if you look at the paper, the first supplementary figure, we characterized the assemblies. We did even mass spec to show that each form has, on average, 1 fluorophore. So each alpha-synuclein has 1 fluorophore. We are really very straightforward for this, because I am aware that if you add 10 fluorophores to one molecule and one to another, it could be that what you are observing is the consequence of the fluorophore. And these fluorophores are sometimes sort of hydrophobic.
So what is that white black
Uh-huh.
And how long does the fluorophore survive?
It survives pretty long, depending on how much you inject. I think it survives all the time because these aggregates are not degraded, and this is what I'm showing you, or slowly degraded. But at one point, we can't see them anymore because, in fact, they are spread all over the place, and we have, a threshold for microscope. This doesn't mean that they are not really anymore here, although that's what this experiment is showing. They are cleared to lower than the threshold of any method we can use. So what we did here is we injected the aggregates in the cortex, and we looked at them in the striatum or the substantia nigra, and you can see that they are found in the striatum and the substantia nigra. And I would say that this is sort of the background because this is the control, and they are, they are above the background. Then we subjected them into these tissue to limited proteolysis, what we have been doing from the beginning as a sort of tool to see in what conformation they are. And you can see that the fibrils resist proteolysis up to at least 0.1 microgram per ml of proteinase K, while the ribbons are degraded as we were expecting they would be. Now, we looked also, after injecting the animal, at what is aggregated, because here we are looking with a pan-anti-alpha-synuclein. Is it the exogenous material? It's exactly your question. Is it the exogenous material that we are detecting, or is it the endogenous material? Because if it's the exogenous, well, then it's interesting, but But if it's the endogenous, this means that we are recruiting the endogenous material, alpha-synuclein. We are crossing a species barrier, exactly like for prions. And this is the result. So this is the input material that we injected to the animal in a monomeric or fibrillar form. These are native gels, that's why they look awful. You have smears because it's fibrillar material and aggregated material. This band, you don't care about this band. This is like a non-specific band recognized by the secondary antibody in our Western blots. But if you look with a human alpha-synuclein, we are injecting human alpha-synuclein to these rats, you can't detect it. What you can detect is a rodent alpha-synuclein that is aggregated. That's why you find it in the slots, and that's why it smears. And it's recognized with a specific antibody that does not recognize the human alpha-synuclein. So the conclusion here is that we injected a human material, fibrils or ribbons. They persist to different extents in the animal, and they recruit, in particular the fibril, the endogenous alpha-synuclein, meaning that it's propagating the same structure, and it's crossing a species barrier. So then, although we are playing with fire here, we said, okay, so we are injecting in the brain and so on. What happens if we inject in the bloodstream? So that's what we did. We had repeated injection of alpha-synuclein in a fibrillar form, oligomeric ribbons, etc., in the bloodstream of these animals. We had 2 injections per month. The fibrils are labeled with a red fluorophore. It's Atto 550. And just before sacrificing the animal, we injected the soluble form of the protein, labeled with a fluorophore that fluoresces in infrared. This allows imaging what's happening in the animals, so you can see that as soon as you inject this material, it goes to the brain to different extents. Then we looked, after killing the animal, at where the material is. And what we saw is that we have deposit in cortical neurons. And what is really interesting is that, remember, we injected fibrils labeled in red, 550, and we injected the soluble form labeled in the infrared fluorophore. And what we saw is that not only the exogenous fibrils or ribbons are sort of precipitating into the brain or taken up by the cells, but the second sort of shot of soluble protein is being recruited and is co-aggregating with the seeds we placed here at the beginning. And if you look at inflammation, you can see that there is mostly inflammation here in the thoracic spinal segment with ribbons and fibrils, and mostly with fibrils. So to conclude here for these studies, what we observe repeatedly with many models from cells to animals is that the fibrils are extremely toxic. So these polymers are made from exactly the same protein. It's not mutants. We have 5 different forms. They look all more or less fibrillar, but they look different. They look different in the electron microscope, although fibrillar. They have different molecular barcode. It's the proteolysis pattern. They have different flash codes. If you want to learn something about this form, you just flash this. They look like, because they are made of exactly the same material, alpha-synuclein, they look like spaghetti for these guys, linguine for those. We didn't have any yet, so I would like to have some before. Maybe you are thinking not to get me alpha-synucleinopathies, but I would appreciate And these are yielding different strains. And so if you want to go further into these studies, I presented 10% of what is in these papers. This one has, the one that is appearing later on today, has 10 supplementary figures with panels from A to Z, and so on. That's right. And you will see that in the paper we duplicated the experiment. We did the experiment with AAV expression or without AAV expression. So we overexpressed alpha-synuclein to force the system, and you will see that, for example, the usage of the foot is greatly diminished when you overexpress alpha-synuclein. But I chose not to present the AAV because I was thinking that someone would just bump up and say, but this is totally irrelevant, but whatever. So you will see that everything is done with or without. So just to conclude, is this specific to alpha-synucleinopathies? My answer would be no. Let's get back for 1 second to Huntington. Huntington exon 1, I can assemble into fibrils or things that look like sort of amorphous. These different objects bind to different extent to cells. We can use, we can also assemble these Huntington in the presence of small amount of HSC70. HSC70 for Huntington does not interact with the fibrils, so it doesn't change their properties. But still, if you look at using a spectroscopic method, it's called Fourier transform infrared spectroscopy. You can see that the structure, the amyloid content of the fibrils made without or with HSC70 are different. They look the same, but they have different abilities to seed the endogenous protein that we express in our reported cell line, meaning that it could be that for other protein as well, you could have different strains, depending on the conditions that you are using. And again, this is from my lab, but other people also published that there are strains. Just at the same time that we were publishing our Nature Communication paper, the lab of Rob Tico in the States showed that if you take section from diseased people developing Alzheimer's disease and amplify the plaque that are in there in the presence of recombinant A-beta or synthetic A-beta and do solid-state NMR, you see differences between different patients, meaning that it could be that, again, patient 1 and patient 2 don't have the same plaques. That's why they are amplified. And you can't see the plaque, the original plaque, because they are not labeled. Solid-state NMR, you label your protein with N15 and C13, and that's all you see, okay? So what is the input is absent, so it doesn't depend on the input.
This was my comment, actually, that I'm a clinician, right? So I see that patients with Parkinson's disease are different. We can never predict, when they become demented or not. So can it be because, the pathogenetic or the nasty power of the Alpha-Synuclein is different because they're different?
Exactly. So that's why I wanted to thank you for this question. I don't think we should at one point revise the term Parkinson's disease. I think it's Parkinson's diseases and many diseases. We were lucky enough with the work of people like Heiko and others to be able to distinguish to some extent, for example, DLB from the rest. At one point, I'm pretty sure that like a century ago, they would put everyone in the same like pot. Now we are starting to distinguish MSA, 2 forms, DLB, probably several forms, with or without other aggregates, and probably Parkinson's disease is going to be a multitude of diseases. Now, having said this, I mean, if you read carefully this paper, there are problems in this paper, because depending on whether he took the aggregates from the occipital lobe or from the temporal lobe in the same patient, he had different spectra. So it could be also that we are in this experiment doing something wrong with the amplification. Okay, but this is again not the only example. You heard that the aggregation of tau causes different diseases, and Mark Diamond assembled a fragment of tau. It's not full-length tau, please. Don't even though they call it tau, it's not tau, it's a fragment. He assembled it in test tubes, and he was very clever, I find. He used these recipient cells that are expressing tau to select, like sort of to sort the different aggregates that he was producing based on the kind of aggregates that the exogenous fibrils were inducing in the cells. And he had a number of classes, some with like enormous blobs, some are with very, very small blobs. He purified this, re-injected them into the animal, and he saw different patterns. So I encourage you to look at this paper as well. So what I told you is that essentially all of these proteins resemble prions. These are in constitutive protein that aggregate into insoluble particles that resist to different extent the clearance machinery, meaning that in some cases we can get rid of some of these aggregates, in some cases we can't because they are so resistant to proteolysis. They amplify by breakage. They pass from cell to cell. There is a toxicity that could lead to neurodegeneration. They occur spontaneously, although there is a strong dependence on aging time and polymorphism, point mutations, or overexpression. I hope I convinced you that there are strains that differ structurally, because I would be happy if one or two people here accept that, structure could explain a pathology. We still have a long way to go. That's exactly what Maria Gracia was saying. We need at one point to show that what is happening in distinct patient is indeed different. I showed you that you could contaminate an animal, which raises a number of concerns. So I'm cautious, but at the same time, I think it's not a good idea to play it the ostrich way. In other words, find a hole and stick your head in it. So that's why in my lab, for example, we manipulate with caution all of these proteins, especially that we are manipulating grams. So we wear masks, we wear eyeglasses. It's forbidden to sonicate and produce aerosol, because remember that our olfactory bulb is exposed like the animals. We wear gloves, lab coats is obligatory. The gloves are outside the lab, you cannot get in. And we implemented cleaning procedures. I can't speak about this at the moment, but we do have procedures to inactivate alpha-synuclein fibrils. And I wanted to finish by thanking the funding agencies who are supporting us, thanking my co-workers, and in particular, this guy, he's here hiding, that's Luc Bousset. He's a great person, brilliant. And I would like to, again, insist a lot on the fact that none of what I showed you would have been possible without a wonderful network of interaction. It's a sort of populated network, but it's fun because you learn from others, and I hope that they learn from us. And I would like to point out the major collaborations we have. Ron Copito and Michel Braic in Stanford. Patrick Brundin, who is now in Grand Rapids, but he used to be here in Lund. I would like also to point out the structural collaboration for solid-state NMR with Anja Buchmann and Beat Meyer, who's the god of solid-state NMR. And I would like also to thank Veerle Bekkeland from the Catholic University of Leuven, with whom we made a major step to move from in vitro with our cell cultures to in vivo, to sort of close to the real world. And I would like to thank you for your attention, and I hope you have questions.